So we have a lot of collaborations with the military hospital, Eliava, UZ Ghent, UZ Leuven, and also Comate, which designed our machine. Actually, why I'm here is about this picture. This is my mom 10 years ago in the clinic. She had a melanoma, a huge one, and she had a lot of chemotherapy. They were in the end successful. And just when she was about to be released, she obtained an AMR infection. And the only thing that could be helped was the vancomycin. But now there are already vancomycin multi-resistance bacteria also. So fortunately, my mom still lives. And that is why we are now here in a phage therapy meeting. And it's all about phages and the use of phages in therapy. In that endeavor, if you want to do phage therapy, you have to know the quantity of the phages in your sample. And actually, there is only one method which is accurately measuring those phages, and that's double agar overlay method. All the other methods are a derivation of that and are less accurate. You can also go to virus counting and qPCR. And actually, our question was, how can we make this method more simple? So the double agar overlay method, you all know it, is the golden standard for over 80 years. You have an accurate quantification. You get a number PFU per ml, plaque forming units per milliliter. And it's used in discovery as well as in QC in industry. But the downsides are that it's time consuming and it's very labor intensive. You have a low dynamic range and you have a lot of waste. So how can we improve quantification of phages was our main goal. We resorted to qPCR because we were really good in quantification by PCR. We have a lot of bioinformatic experience. And for the BFC2 cocktail, I always pronounce it wrong, from the military hospital, we designed a platform to get an accurate quantification of phages. And we also did interperson validations between the double agar overlay method and the qPCR method. However, there was a discrepancy if you looked to the qPCR results and to the DAO results. You had always more qPCR, more phages if you looked with qPCR than if you looked with double agar overlay. And it was depending on the type of phages. So some phages did give a huge variation or a huge amount of surplus, and others gave less. So why did we find more phages in qPCR than in DAO? In essence, if you do qPCR, you count molecules. And in DAO, you count infective phages. So we could solve that. The DNA found outside of the phages, colored with antibodies which have a gold particle, could be found outside of the phages, and this is results with electron microscopy. So we could partially clarify the discrepancies. So we were back to off. We wanted to improve quantification of infective phages 2.0 with a less laborious method. And in essence, first thing we wanted to do was design a proof of concept. Here you see a movie. You have here a droplet of 200 nanoliters with bacteria and phages in it. And here you have a droplet with only bacteria in it. And you can see that there is a difference after a certain amount of time. It's kind of turbidimetry. We also designed a software package for it. So, but this was the start for delving deeper into the microfluidics. We got to experts and they said, this is impossible to do. 6 years later, we ended up with microfluidic phage quantification. So we have a chip, we have a device, and we have software. And in the chip, in essence, you can see droplets and those droplets are dark if they only contain bacteria. And if there is one phage in a droplet, that phage will destroy the bacteria population, and you will have a clear droplet. So we had a lot to optimize: imaging, incubation, microfluidics, temperature, and so on. For all the black things, there are, I think, 5 other parameters we had to optimize. But we end up in a device we automated, and with bioinformatics, we can now solve the solution in a really rapid manner. So we have the microfluidic chip. It consists of 2 simple inlets. You add in one inlet a phage and a bacteria solution. You add in 2 other inlets oil. And here is an incubation chamber. And then you have an outlet. And here we show an image of the incubation chamber filled with droplets. Each droplet is circled with a green circle, which is then evaluated in function of time. We get around 2 billion data points during the experiments, so we need a little informatics to solve that. The device is literally a black box. It has a drawer. We put our chips in that drawer, we close the device, and everything then is fully automated. The data is sent to the cloud, and it's analyzed, processed, and we get our results out of that. So to recapitulate, in the device, we get droplets. The droplets are incubated in the device itself. We have imaging analysis in that. And then you can see the image like this. Each droplet is followed individually. And I just cut out of this range 2 droplets, 1 droplet with bacteria in, and 1 droplet with bacteria and 1 phage. And I'll try to show what happens in function of time. Both droplets start growing, and after a while, the population in here disappears because of the phage, which destroys the bacteria, the living bacteria. So you really look into one cloned phage, which does destroy that population in that drop. So you do that plus 10,000 times, and you end up, this is again a piece of the image you saw lately. And what you see is a cloud of bacteria and phages, which is clear, and a bacteria droplets, which are with a higher grayscale. So here you see grayscale, and you see the position, the exposition of the droplets in the array. And we can look at it in various ways. So what we have as an output, we have the first output is susceptibility. Is this phage really going to be able to destroy this bacteria? Yes. And you have, as a surplus, the quantification, which is rather accurate because we do also statistical analysis to avoid that there will be 2 phages in 1 droplet. So we correct for that. And this experiment was done on Salmonella typhimurium, and it took around 3 hours to get the final result. So we started looking into more bacteria phage couples. First, we looked into the escape set with different phages. And here I show just an overview of those escape pathogens. And you can each time see that you have those differences between bacteria grown in a droplet and bacteria which are lysed. Each time you see different aspects in the droplets, but we managed to evaluate each type of difference. We also went a bit further because the first thing the clinicians said to us, but you did not, you only looked to lab strains. So we now have a whole set of clinical strains for instance, Pseudomonas aeruginosa to test with their respective phages. And we also looked in foodborne pathogens and also plant pathogens, and actually animal pathogens. I resort them as with the human ones. So each time we can see differences between phages and bacteria. And one of the last things I'm going to show is a thorough validation study of the methods. So what we did here was we had a sample of Salmonella typhimurium and phages, and we checked whether what we would come out with the double agar overlay methods. We did 8 experiments. Those experiments were done in Duplo with 2 different people, and we got a coefficient of variation of 44%. If we then do the droplet experiments with cultures, bacterial cultures, we cultured overnight, we get a variation of 23%. But if we go to making droplets with logarithmic cultures, we end up with a coefficient of variation of 4%. So that's well below the industrial accepted standard of 15% you need for a biological assay. And it looks as if we report a higher concentration, but that is probably because here in the double agar overlay method, maybe phages will be attached to that bacteria in that population. That we do not know for sure yet. We can do some experiments for that to look into that further. But we seem to have a fairly robust method that could be used. I think the most important thing is that we have a 10-fold higher precision. It seems universally applicable. I always say we have 59% success rate. And we have at least 10-fold less labor to perform to get to our result. And the top other things are that it's an automated system. If we would send the machine to you, wherever it is, in Antarctica or in Brussels, it would give the same results. And you do not need any microfluidic experience because we handled all the microfluidic problems in the machine itself. Future perspectives are that we want to further validate the machine on relevant clinical samples and relevant clinical bacterial samples and phages. We'd like to expand our throughput with more samples in the same time. We are now looking to OEM integration for fluorescence or luminescence. You already saw people do more rapid tests, but we specifically designed now for a universal applicable machine, because whenever you go to fluorescence, it will end up in a lot of optimization for a particular strain, and that can be done. It's not a problem at all to change the imaging system in our design. One of the comments was also you work in fluidic conditions. We are nearly there to do it in solid phase, and we are open for collaboration. So to remember, it's an universally applicable method. It's standardized in contrast to the double agar overlay method. It's less laborious and it's more precise. Thank you for your attention.